Okay, looks like we lost some people over the three-day weekend. I hope you all had a nice weekend. And let's go ahead and begin with just a little announcement that I was asked to make. And this slide is also posted in along with the lecture on the website, so you can look at it there. But it's about a student health outreach club. And if you're interested, you can contact the email address which is shown on this slide. So let me now begin with a brief review of some of the topics that we covered in the last lecture, and then we'll move on to the content of today's lecture. So we talked about how species arise, and it's really pretty simple. The primary cause of speciation is long accumulation of genetic change as a result of geographic separation. If you separate populations geographically, they can't exchange genes. New mutations will arise independently in the separated populations. They'll likely be different in some environmental aspects as well. Natural selection will adapt to those different environments, cause adaptation to those different environments. Over long periods of time, populations will diverge. That's the allopatric model of speciation. And we talked about two sort of common ways that would occur. One is through the dispersal of a subset of a population into a new isolated environment, like colonizing an island as an example. And the other was the so-called vicariance model, where there is a discontinuity in the environment that separates the two populations, two parental populations, something like a big river that they can't cross, a mountain range, whatever. But those are the common means of speciation. Then we talked about two other more special cases under the heading of sympatric speciation. Sympatric speciation is just meant to refer to speciation that occurs when populations are occupying the same space, or at least overlap in the same geographic region, and therefore there is the potential for genetic exchange. And we used a nice example, the apple and hawthorn flies, each of which had specialized on discretely different resources and began to adapt differently, despite the fact that this had happened over a period of only 300 years since the apple was introduced into North America. Then we talked about polyploidy as a major cause of sympatric speciation in plants. Polyploidy is where there is a doubling of the chromosomal content. An example is a tetraploid. A tetraploid has 4 copies of the each homologous chromosome, rather than 2 in a diploid. If you cross a tetraploid to a diploid, you get a triploid, which is non-functional and sterile. I mentioned the navel orange as an interesting example of a triploid. That's why it doesn't have seeds, because the unequal segregation of chromosomes to the poles in a triploid leads to unbalanced gametes, which are not fertile when you have the union of gametes. You end up with zygotes that don't survive because they're missing some genes, have others duplicated a number of times. So polyploidy is a really important case in plants, and it's a good example of sympatric speciation. If I asked you a test question about sympatric speciation and I listed polyploidy as an example, you would immediately get the right answer, right? So that sort of concluded a discussion of the mechanisms of speciation. We then talked about what happens when isolated populations come back into contact and potentially If there are no pre-zygotic isolating barriers, they could potentially hybridize. Of course, if there are pre-zygotic isolating barriers, they can't hybridize. If they come back into contact, they still evolve independently from the standpoint of the possibility for gene exchange and the accumulation of mutations. If they do hybridize, you can have several outcomes. You can have hybrid zones, which may be transient. We saw the example of the Townsend's warbler and the hermit warbler, and their hybrid was an example drawn from the textbook. If they hybridize and produce an inferior hybrid offspring, there's likely to be reinforcement selection against hybridization because the reproductive potential of the two parental populations is reduced by hybridization if the hybrid offspring are inferior, less likely to survive and reproduce. So these are some of the consequences of isolated populations which have become essentially different species coming back into contact. We then turn to talk a little bit about how you construct phylogenetic trees. We just touched on that subject. Books and books are written on the subject of constructing phylogenetic trees. It is a complicated area. In fact, this idea of how you infer a tree or a network is an area of mathematical investigation. So what we did was just to give you a tiny sense of the flavor of this subject in a few minutes. And that's all you're really expected to know for the purposes of this class. We introduced the fact that there are two common approaches. One are called phrentic approaches. They're based on a matrix of pairwise similarities or distances. Generally, you might think a common measure would be percent similarity. Let's say we do something specific. We sequence a gene. We take 4 species, and we take a particular gene, and we sequence it from each of the 4, and then we measure the percent of bases, which are similar between the two. That's a percent measure. It's a continuous measure, goes from 0 to 1. We then just cluster those pairs based upon those which are most similar using a clustering algorithm. What's an algorithm? It's a set of rules for carrying out some mathematical or statistical operation. So we use a clustering algorithm to build up clusters based upon their similarity with regard to this percent sequence similarity in the gene, as an example. So that's one area of, for the inference of phylogenetic trees. The other is different conceptually. The other method that we talked about are called cladistic methods. They're different because they emphasize shared ancestry between discrete traits. So rather than being a continuous measure like a percentage, we're working with a discrete measure, sharing a particular mutation, for example, in the DNA would be a discrete measure. Another would be sharing some morphological similarity, for example, some bone structure in the hand between, let's say, humans and some other primate group, whether you share that discrete structural difference or not. Sharing it is taken to imply common ancestry, okay? A common ancestor, derived from a common ancestor. Now, it turns out, there is a mathematical formula for the number of trees you can get if you have in different taxa that you want to build a tree of, okay? It's actually a factorial expression. The number of trees that you can get from even a small number of taxa is really large. It grows much faster than exponentially. So when we use these cladistic methods, typically there are not as many characters as there are potential trees if we have a large number of taxa that we want to organize into a tree. What does that mean? That means that we will have several trees which will satisfy the criterion of parsimony, and that criterion is that we want to minimize the number of character state changes over the tree. That's the best outcome for cladistic trees, is to minimize the number of character trait changes implied over the tree. But that will not, usually it doesn't give us a unique outcome. There are usually a number of trees which will satisfy that criterion. So there are two methods. For the purposes of this class, there are two methods that we've talked about for inferring phylogenetic trees. They're conceptually different. One is the Frintic method, which is based upon a clustering algorithm. The other is the cladistic method, which is based upon shared derived characters that imply common descent. And you look at a larger number of characters as possible to infer a tree, and you do it in a way that minimizes the number of changes that could have occurred over the tree. And then there are words that are introduced to describe things. One of the things about science in general, biology in particular, is that it's often important to invent terminology because new terms capture a meaning of a concept or a description of a phenomenon in a single expression that conveys a whole chain of reasoning. So, and this is probably true for the evolution of language in general, but in this area there are a number of words that get introduced. One is synaptomorphy, that's shared derived characters. Another was homoplasy and homology, okay? If you know those three words and what they mean in the context of building cladistic trees, I'll be happy. We then went on to talk about the limitations of methods for phylogeny inference. We talked about homology. This is where a trait is derived from a common ancestor, versus homoplasy, where there's been convergence. The trait actually has become similar due to convergent evolution. And as an example, we considered the dolphin versus the ichthyosaur. You know, remember the pictures are beautiful when the two look the same. The dolphin and the ichthyosaur look very similar, even though the dolphin is a member of the mammal family and the ichthyosaur is a reptile. They have evolved convergently because they have adapted to a similar ecological niche. They're marine animals. They're adapted to swimming with bodies that are agile, long jaws, sharp teeth, very similar, but they have a completely different evolutionary history. Now we're going to look at another example, and this is the whale. And also, this is also drawn directly from your book. And here, we're going to see where traditional phylogenetic trees have actually been incorrect in their inference about where the whale comes from, what its correct placement is in the tree of life. Whales are also marine animals, and they actually belong in a group of mammals called the artiodactyls. But one of the key traits that define the artiodactyls has to do with which are mammals with hooves and an even number of toes, and they have this unusual pulley-shaped ankle bone called the astrologus. Well, whales lack that. And if you look at the classic phylogenetic tree, because of, this is a figure straight out of your text, you see the picture of the astrologus, which provides this key character which is used to infer common descent. Based on that, the whales are placed outside the archaeodactyls, which include the camels, peccary, pig, hippo, deer, and cow. But if you look at DNA-based or gene-based evidence, you get a very different picture. And that's shown in the bottom of the slide. In this case, what was initially used were the presence of elements in the DNA called sign elements. Now, it happens that these are retroviral derived sequences that are mobile, that occasionally in episodes over evolutionary time invade a genome. And you see that the whale and the hippo share a series of 4 different sign elements, implying a common ancestry, a close common connection between the whale and the hippo. So sometime in the lineage that preceded the separation of whales and hippos, this sign family invaded the genome of the common ancestor, and those are still shared between whales and hippos, grouping them together. Well, which piece of data do you believe the most? Do you believe the astrologous data, or do you believe the DNA-based data? Well, subsequent researchers have sequenced genes, and when you do that, there's no doubt that the whale is most closely related to the hippo. So this is an example of an error in phylogenetic inference because of the loss of a character, the astrologus, in the evolution of the whale. And the true picture is a whale most closely related to the hippopotamus. So that's another example of the kinds of errors that can arise in building phylogenetic trees. But as we accumulate more and more data, and particularly if we accumulate more and more DNA-level data, we converge closer and closer to an accurate picture of common ancestry. Now I want to talk just a little bit about the fossil record. The fossil record is really important because it tells us most of what we know about the organisms which are present on the Earth in the past. It's a physical trace of organisms that lived in the past, and the total collection of these traces is the fossil record. Now, an important thing to know about the fossil record is it's the only direct evidence that we have of organisms that lived in the past. Unless we can invent a time machine and go back and look, the only direct evidence we have is the physical traces that still remain in the geological formations that we recover fossils from. And this tells us quite a lot about what lived in the past, what it looked like, and possibly how it existed. How do fossils form? Well, they typically form in a way like as depicted in this slide from your book. You have organisms which are living close to a site of potential deposition. This is often along a river, an estuary, a place where their remains are likely to be buried. So we consider, for example, this tree. Its leaves and pollen and seeds are going to fall into a nearby stream or river, get carried to a site of deposition, buried by silting. As time goes on, the sediments build up and the remains that are deposited become compressed over millions of years, and we finally end up with compression fossils. Fossilization is a very rare event. You've got to be in the right place at the right time to leave behind fossil remains. It's very rare. Your book has a kind of a neat calculation that I found very appealing, and this is about Archaeopteryx. Now, many of you probably know that Archaeopteryx were among the earliest flying reptiles. They're clear evidence of reptiles adapting into bird-like organisms. And we have a relatively small number of Archaeopteryx fossils. It happens that actually most of the fossils come from a particular site in Germany. I think we have about 10 or 12 fossils at this time of Archaeopteryx. Well, your book does kind of a nice calculation. It says, well, now let's suppose that the average size of the population of Archaeopteryx was about 200,000 a year, and that they persisted for the average period that species persist, which is 2 million years. So that's 200,000 times 2 million, or 2 to the 5th times 2 to the 6th, or 4 times 10 to the 11th, right? And we've only got about 10 of them. So the chance of any particular one of those Archaeopteryx getting deposited in the fossil record was only about 1 over 4 times 10 to the 10th, right? 1 over 40 billion. So the chances of any particular organism ending up in the fossil record are very small. But because the species persisted for a long time and there were enough individuals, by chance, a few got deposited, died under conditions where their remains were preserved for millions of years, and that tells us about the physical existence of Archaeopteryx in the past. Otherwise, we would not know. There are different biases, though, about how material get deposited in the fossil record. One is habitat bias. That just means the population has to live in a habitat where there's a good chance of ending up in a potential site of deposition. Okay? If you live in the desert or on a mountaintop, you're less likely to end up in the fossil record than if you live on the floodplain or a delta. Habitat bias. There's taxonomic bias. Organisms with bone structures or calcareous shells are much more likely to be represented in the fossil record than are organisms that lack those features that are easier to preserve through time. So that's taxonomic bias. There's temporal bias. Some recent fossils are more likely to be found than older fossils. Remember when we were talking about Neanderthals, I said that they got into Europe somewhere between 350,000 and 600,000 years ago. We don't know any more precisely. Why? Because most of the Neanderthal sites of habitation in Europe were destroyed by successive periods of glaciation. So the ordinary processes of time, on a longer time scale, subduction of continental plates will destroy any fossil record that was contained in the rocks that are in the material which is subducted. So the longer the period of time, the more likely it is that the fossils will be destroyed and we won't know about it. So that's temporal bias. And then finally, there's abundance bias. Abundant organisms are going to be more likely to be represented than rare ones, just because of the, like the calculation I did on Archaeopteryx. If they're more abundant, there are going to be more fossils. How do we date fossil material? Well, we've got 3 ways that we can approach the dating problem. The classic method, the method that's been used since early in the 19th century, is the analysis of sedimentary layers. Now, because of this deposition process, we know that layers in the strata, which are closer to the surface, are going to be younger than layers which are further from the surface. So that gives us a relative dating, but it doesn't tell us anything about absolute time. But we can use the sedimentary layer as one kind of relative dating. We can get information on absolute time from radiometric dating, which we've been now using ever since the 1950s. In fact, Willard Libby, who pioneered that technique, won the Nobel Prize for developing carbon-14 dating. And then in more recent years, there's another kind of dating that we also use, and that's called a molecular clock. And that's based upon simply counting the number of mutational differences between two related lineages. The number of mutational differences is a measure of the time since the separation of those lineages. So there are three approaches that we can use to timing, and they're independent. They're quite different. One is the sedimentation, the sedimentary layer. One is based upon radiometric techniques, and the other is based upon the accumulation of mutations between different lineages. Your book has a wonderful folded several pages that show this wonderful picture of the geologic record, and I hope you'll spend some time looking at it. And it begins with the origin of the Earth and goes through to the present, and on one scale it talks about the geologic time scale, that is the one we infer from sedimentary analysis, the classic time scale, and then on another column, it gives you the absolute times that we've been able to infer from radiometric methods. And it shows the different major groups of organisms or the big events in geological history that occurred through time in these different epochs. So please look in your book at that, because it's well worth studying. What I've put here is just the absolute time, mostly inferred from radiometric methods, against big events in evolutionary history. The origin of the Earth, 4.6 billion years. Earth cools and solidifies by 4 billion years ago. First evidence of life, at least 3.6 billion years ago. That's really amazing. I mean, it's quite remarkable. We're going to talk in a few minutes about how life might have originated, but it didn't take long. It gives you something to think about. It turns out that one of the big things that's happened in astronomy over the last 20 years is the ability to use both indirect and direct techniques to identify planets around nearby stars. This is something we couldn't do until about 25 years ago. And we now know that there are literally hundreds and hundreds of planets that we can determine in the nearby parts of our own galaxy. And recently, in the last couple of weeks, there was a publication on the number which are, quote, Earth-like in the sense that they potentially could harbor life. Well, there were 40 or 50 that fall into that category, and as time goes on, that number will grow and grow. So the universe must contain a huge number of other Earth-like planets where life could evolve. But this is how it happened here. It started fast. Within 400 million years or less, there's some estimates that life began 3.85 billion years ago. Really amazing. So that's the first evidence of life on Earth. Then the oldest prokaryotic fossils, 3.5 billion years. Remember, I mentioned in an early lecture that one of the big events in the history of Earth was the change in the composition of the atmosphere because of life. Life itself changed the composition of the atmosphere through the evolution of photosynthesis. Photosynthesis produces oxygen as a byproduct. This led to an oxygenated atmosphere. Our distant ancestors had to adapt to the use of oxygen. The primordial atmosphere had no oxygen. So accumulation of oxygen, roughly 2.5 billion years ago. Origin of eukaryotes, that's the lineage we belong to, about 1.7 billion years. Oldest eukaryotic form, about 2 billion years. Origin of multicellular organisms. Big step in evolution to have this, somehow, evolution of cooperative behavior between different cells so that they could combine and be more effective working together. It's the beginnings of multicellularity, one of the great steps in evolution in the history of biological diversity on Earth. Oldest living animal fossils, only 750 million years. Plants and fungi colonized the terrestrial land only 440 million years ago. It's pretty recent on this time scale. And then the extinction of dinosaurs, which we'll come back to, 60 million years. First humans, maybe 200,000, 250,000 years. So that's the picture of the history of life, the geological history of life on Earth. Now, it's one of the things that's kind of interesting that's become again, a subject of scientific investigation in my scientific career, not in the last 100 years, but in my working lifetime, has been the question of extinction. We know now there's two kind of general ways to look at extinction. There's what is called background extinction. I mentioned that species, on average, persist for about 2 million years and they tend to disappear. That's pretty much a continuing process. Earth changes, adaptation proceeds or it doesn't. Some species become less successful than others, some disappear. But there are also episodes of mass extinction that we can now identify from the fossil record. In fact, there are 5 episodes of mass extinction since life colonized the land, just in the last 500 million years. 5 episodes of mass extinction. One of them, the Permian, the end of the Permian extinction, killed more than 90% of all the life on Earth. Bang, out in the... So how do we define a mass extinction? Well, we say more than 60% of species are gone in a million-year time frame. In the blink of an eye in evolutionary time. 90% in the Permian extinction. And more recently, about 65 million years ago, we had a major extinction event which we know quite a bit about. We don't know much about what drove these earlier extinction events. We can only speculate, but we now know quite a bit about what happened 65 million years ago, because it's almost certain that a large meteor or asteroid struck the Earth. This was 10 kilometers in size. It's huge. 10 kilometers converts to a little over 6 miles. It's a huge rock. This thing was as big as Mount Everest is high. And it struck the Earth about 65 million years ago and caused the extinction of 60 to 80% of all multicellular species. All right. Now, how do we know that? What's the evidence to support that? Well, there's several kinds of evidence. One is that the iridium is very rare in Earth rocks, but it's common in meteorites. And we can show that there was a sudden splash of iridium over a wide area that dates back by strata to about 65 million years ago. A second is direct evidence of a large impact, or what are called microtektites. These are where silica, sand grains, have been melted by huge impact and heat and then recongeal into structures called microtektites. And we can show that they're very common at that time horizon as well. Finally, it was possible to identify a large impact crater just off what is now the Yucatan Peninsula of Mexico, and to show that the impact crater was associated with a big increase in microtektites, and that the material splashed out of that region was high in uridium. And by the way, the asteroid struck at an angle and caused this huge splash of material all the way up into North America. So it's an incredible event, and it led to huge changes in the whole global climate because of the amount of material which was dispersed into the atmosphere, which blocked sunlight, years of effectively a winter caused by the blockage of sunlight by this material which was circulating in the atmosphere, and the extinction of a large number of species. Some species survived, obviously. We're here, we wouldn't be here. And in fact, one of the interesting things is it was selective. Okay, so this figure just shows you the jump in iridium at 65 million years as evidence of the asteroid impact. Here's where it presumably struck around the Yucatan Peninsula. And this is the imagined splash of material out from the asteroid striking. So some lineages apparently were better able to survive this catastrophic event than others. And our ancestors, mammals, were among them. But the dinosaurs, flying reptiles, and so forth, they all went extinct. And it isn't clear what the differences were that allowed mammals to survive while the dinosaurs went extinct. But, and people are trying to understand better, to create better testable hypotheses about why the event was selective in terms of the forms of life which survived. So after the asteroid impact, life recovered. You know, life is very tough. I mean, we're going to see when we talk about bacteria and archaea that life exists in habitats we wouldn't imagine are habitable. So life is very tough. You really can't very easily sterilize the Earth, and this event certainly didn't do it. Within 4 to 6 million years, all of the major families of mammals had emerged and had diversified into the sort of ecological niches that had previously been occupied by reptiles. So mammals became very successful as a result of this catastrophic event. And so that's fossilization, and extinction, and what we know of, just a taste of what we know about the past record of life on Earth. Now I'm going to talk for the rest of this lecture, and probably for part of the next one, about bacteria and archaea. Remember when we talked early in the class, I mentioned the fact that the lineage which we call archaea was recently discovered within the last 30 or 40 years by a remarkable scientist named Carl Woese. It happens that on Monday, the LA Times had an obituary for Carl Woese. But one of the major lineages of life has only recently, relatively within my career, been discovered. That's because we have been able to use DNA-based and RNA-based methods of inference to reconstruct the history of life. And that revealed that there were two different kinds of, broadly, of single-celled organisms out there. One was the bacteria as we know them, prokaryotes, and the other was the archaea. Also revealed the archaea are actually closer on the evolutionary lineage to eukaryotes than they are to bacteria. Now, if we look at this tree, down at the bottom there's this common ancestor of all species living today. And I've told you before that the evidence is that all life today had a single common origin on Earth. One of the strongest pieces of evidence is that all forms of life use either DNA or RNA as a hereditary molecule. very likely, this is an application, by the way, of the shared derived character idea, that we all use a common system for information transfer, all life on Earth does. But how did life arise? And what is life? Well, for the purposes of our discussion, we can say that life is just a self-sustained chemical system capable of Darwinian evolution. Okay? Self-sustained chemical system capable of Darwinian evolution. And with that definition in mind, we then can try and construct hypotheses about how life might have emerged on Earth. And there are three broad categories of possible explanations. The first is that it arose through natural chemical processes that we can investigate scientifically today in the laboratory, that we can erect hypotheses under this assumption and test them. That's sort of in an intellectual sense, it kind of is a natural outgrowth of the old uniformitarianism idea that we can observe through what we know about the principles of chemistry, what might have led to the origin of life. Another possibility that gets discussed sometimes is that it came from outer space. Well, who knows, maybe life came from outer space, but that doesn't really solve the problem because the question of origins just gets moved back to some other point. Or the third is some sort of supernatural, special creation of life on Earth. That's not a scientifically testable hypothesis. So we'll focus on the ones that we can test scientifically, that life arose from chemical processes. And if we do that, then we can make some deductions. The first thing that we can say is that if that hypothesis is correct, then the predictions that we draw are going to have to be consistent with physical and chemical laws, right? What we observe is physical and chemical laws. And they're going to need to be consistent with the law of natural selection. That is, they have to be able to produce genetic variation, which can be selected and allow for evolution. Now, the logical deductions based on these principles will exclude a lot of possibilities. If we just stick with those two principles, there's a whole universe of other possibilities that immediately get excluded. So then we just focus on the consistent scenarios, and we try and perform experiments or simulations under those assumptions to see how far we can get. And it turns out that we've actually gotten quite a ways in recent years in understanding how life might have evolved on Earth. And some of the first work was actually work done by Oprin and then later by a graduate student at UC San Diego named Stanley Miller. And he set up a simple experiment called a spark chamber experiment, where they introduced what they thought would have been some of the gases that would have been present in the primordial atmosphere of the Earth. Water vapor, methane, ammonia, hydrogen, carbon monoxide, in an aqueous environment. And they provided a heat source to sterilize this and then and to allow condensed moisture to fall into this chamber of the primordial atmosphere. And they introduced an energy source, a spark. So they just had two electrodes and they would cause a spark to pass between the two electrodes. And after a period, they sampled the contents of the chamber, and they were able to show that they got a number of, the energy input caused a number of spontaneous chemical reactions that led to molecules that looked like some of the precursors of life. We got molecules that looked like amino acids, some that looked like purines and pyrimidines. Extensions of these experiments showed that it was possible to get proteinaceous material produced spontaneously in this kind of an experiment. So this demonstrated that it was possible to synthesize the simple building blocks of life under conditions which were very likely to have been present on the Earth in its earliest days. Now, we sort of fast forward to the present time. There are several theories about what the most important molecules were at the origin of life, but probably we think now that the kind of key molecule was an RNA-like molecule. We call this the RNA world hypothesis, because RNA can be synthesized in these sorts of spark chamber experiments. So hypothetically, it certainly is physically possible that it could have been present in the primordial environments. Secondly, RNA has two essential capabilities. It is actually able to catalyze some chemical reactions. So it has a catalytic ability, and it has an information transfer capability. So we had one molecule that was capable of doing two things. It could have run some of metabolism because it was capable of catalysis, at least in some cases, and it has an information transfer capability. We know, for example, that RNA participates in the catalysis of the peptide bond in protein synthesis now in your bodies. So RNA is an important catalytic molecule. It's also been shown that RNA can replicate itself in the presence of zinc. Now, zinc is found in clays, and the sort of thinking is that in this very primitive beginnings of life that you would have had this soup of material produced by abiotic processes, but it needs to be confined in order for the energetics of the reactions to be high enough. So if it's confined on a surface, like pyrite, like the kind of surface you find in some sorts of rocks, there's a likelihood that you could have had some sort of metabolism begin. You can also show that you get cell-like structures that form more or less spontaneously from lipids. So it's conceivable, we at least have scenarios that tell us something about how life might have started on Earth. So this is what we think today, but this is an area of very active research. And in fact, one of the people who's been a leader in this field, Jack Szostak, got the Nobel Prize just a couple of years ago. In your lifetime, I'm sure that you will have a much richer view of how this might have all started than we have today. Okay, so that's sitting down there at that common ancestor, how it all began. Now we fast forward and we say, well, what do we know about bacteria? Why do we want to study them? Why are they important to us? Well, we know a lot about them. First of all, we know that they're really diverse, and this picture shows you some of the diversity among bacteria. We want to study them because there are many practical ways in which we use bacteria in fermentation, for example, in many other ways, so they're of practical economic value. They're also a major source of human disease. So there are lots of reasons why we want to study bacteria, but here's a picture of how bacteria differ. How do bacteria differ from archaea? Well, bacteria are distinguished by different molecules that make up their plasma membrane from archaea, and they are also different from archaea in some of their RNA and DNA transcription and translational machinery. These are very fundamental. And I'm just about out of time, so I will pick up this topic on Friday. Don't forget to bring your clickers on Friday.